extensive cell renewal. Okay, and this is how you can see that is the bone marrow, which is going to change into the hematopoietic stem cells. Now, where all you find this hematopoietic stem cells? Mostly in the bones, and that is mostly in the pelvis, the femur, the sternum over there, over the rib cages over there. That's the sternum. And of course, even the umbilical cord, you find this hematopoietic stem cells, and some of them is found even in the peripheral blood, because during the blood flow, some of the WBCs are found, not many. You will find them only 1 in 1 lakh cells. You find the hematopoietic stem cells moving around in the peripheral blood. So, of course, the research is also on its way how to isolate those hematopoietic stem cells from the peripheral blood. But otherwise, you usually get it from the different types of the bones over there. So, how to isolate this particular hematopoietic stem cells? So, since hematopoietic stem cells cannot be isolated as a pure population, it is definitely, because this cannot be isolated from just by viewing it under a microscope. They have to be isolated by using a flow cytometry. If you can see in this picture, so that's a flow cytometry, wherein there are some specific cell markers, particularly the CD34 markers. So when these stem cells are used with a little bit of dye which is being used over there, or when it is stained with some fluorescent dyes over there, when it is sent into this flow cytometry, there's a laser beam which is going to pass through this RBCs, and whichever cell is stained. So if you can see among this yellow stem cells, you can also find some blue color cells. These blue color cells are the ones that are being stained by the fluorescence emissions. And these are the ones that is going to be isolated and used for stem cell procedures. Okay, so as I told you, hematopoietic stem cells are not many. You find them only 1 in 1 lakh cells. So they are going to be very, very less, but it is definitely not difficult to isolate them. And the cells that have lost the hematopoietic stem cell nature is called as LIN, L-I-N, if you can see over there. So L-I-N is the name that is given to the cells that have lost the hematopoietic nature, which you find them in a mature blood cells. That's called as LIN. So lack of expression of the lineage markers is used in combination with detection of several positive cell markers to isolate hematopoietic stem cells. So that is how they use this markers, that is CD34 markers, to isolate stem cells. And the hematopoietic stem cell possesses 2 important properties. One is, if you can see in this picture, one is self-renewal stem cells, and the other one is a differentiated stem cells of yours. So the self-renewal and differentiated. Self-renewal is something like a memory cell which has to be stored, because you cannot use up all the stem cells at once and then there is nothing left. So there is some population which forms into self-renewal, wherein this will be used as a stock whenever you want it once again. So that is the reason why they have self-renewal cells and differentiated cells over there. This process gives rise to 2 populations, that is long-term and short-term. Which is a long-term? The self-renewal cells is a long-term hematopoietic stem cell, and the short one is they are going to be there only for a limited period of time, and then they are going to get differentiated. Hence, there is no more hematopoietic nature in them. So that's why they are called as LIN, LIN cells. So the clinical applications of hematopoietic stem cells in the bone marrow transplant is where it is used for aplastic anemia, congenital neutropenia, that is wherein you find only few neutrophils present over there, sickle cell anemia, thalassemia, and replacement of bone damaged bone marrows in case of chemotherapy. So because during chemotherapy, there's a lot of loss of bone marrows in the bones. And the self-renewal attributes of the hematopoietic stem cell ensures long-term hematopoiesis post-transplantation. So this is how you are going to talk about when you're going to talk about self-renewal and the differentiated one. Self-renewal cells, they are long-term hematopoietic stem cells, and whereas the short-term are the differentiated ones. they have their own markers over there. That is, if you want to know the difference between the long term and a short term one, there are some human markers. This is called as DIN negative, CD34 positive, CD38 negative, and so on and so forth. There are different types for the long term hematopoietic stem cell, whereas for the short term, you have very less amount of markers present. So when you're talking about the mouse markers, they also have different types of markers over there to identify the difference between a long-term and a short-term hematopoietic stem cells. So where are the clinical applications applied when you're talking about a hematopoietic stem cells? So this has become a standard treatment for some type of abnormal hematological conditions, which includes multiple myeloma. So what is multiple myeloma? A cancer of the plasma cells is called multiple myeloma. Then you're talking about non-Hodgkin lymphoma, which is called as a cancer that starts in the lymphatic system. Then you also talk about a Hodgkin lymphoma, the beta thalassemia, and sickle cell anemia. What is this non-Hodgkin lymphoma and Hodgkin lymphoma? When you're talking about the non-Hodgkin lymphoma, this can arise in any of a lymph nodes in the body. So we have different places of lymph nodes in our body. You find one at the carotid arteries over there near the neck. Then there are some at the armpits. There are some at the Peyer's patches. If you remember, that is near the intestine, Peyer's patches. Then you have some at the groin region. Then some at the knee, at the back of the knees. You have this lymph nodes. So those cancer that can arise anywhere from the body is called as non-Hodgkin lymphoma. Whereas when you're talking about the Hodgkin lymphoma, this can arise only at the upper body parts, that is either in the neck, the chest, or the armpits. So only there, when you find those cancer cells, then you're calling it as Hodgkin lymphoma. But if you find it anywhere in the body, in the lymph nodes particularly, then you're going to call it as non-Hodgkin lymphoma. So these are the types of diseases that can be cured by this application of hematopoietic stem cells. And allogenic therapy is also used to treat the following disorders. So what are the following disorders over here? You can talk about leukemia. You can talk about acute myeloid leukemia, that is cancer of the blood and the bone marrow cells. Acute lymphoblastic leukemia, that is about the WBCs. Lymphoblastic, it's in the name itself. So you don't have to get confused. What is lymphoblastic? What is myeloid? When you're talking about myeloid, we're going to talk about the blood cells. Remember the myeloid progenitor and the lymphoid progenitor, which give rise to WBCs, RBCs, and this gives rise to WBCs over there. Then you're going to talk about the chronic myeloid leukemia and the chronic lymphocytic leukemia. Chronic is it's going to be continuous one. It is the opposite of acute. Acute is one single time. Chronic is lifelong problem, is what you call it as chronic lymphocytic leukemia, and the myeloproliferative disorders and anemia. Young chronic lymphocytic leukemia patients are able to undergo both autologous and allogenic hematopoietic stem cell therapy. So when you're going to talk about this hematopoietic stem cells, which is collected from the cord blood cells and also from the peripheral blood cells. These are collected after stimulations with the cytokines. That is, this is not available very easily. So what you're going to do is you're going to instigate or stimulate the cytokines such that the granulocyte colony stimulating factor. Remember, granulocyte colony stimulating factor means what? You're going to stimulate the granulocytes. Remember, agranulocytes and the granulocytes. So when you're stimulating the granulocytes with the cytokines, this is going to mobilize the hematopoietic stem cell from the bone marrow to the peripheral blood. So when you want to collect the hematopoietic stem cells from the peripheral blood, what you're going to do is you're going to instigate or stimulate with the cytokines so that these are going to get stimulated and you find more amount of hematopoietic stem cells in the peripheral blood. And in the recent studies, the hematopoietic stem cells from the bone marrow could be mobilized by chemicals also using chemotherapy called chemomobilization. And of course, the current trials have also been used for these cells from solid tumor treatments and for also curing HIVs. Okay, so this is how there are different types of hematopoietic stem cell therapy targeted. So it can be targeted for metabolic disorders. It can be targeted at the cardiac and neuronal disorders. It can also be targeted for hematological malignancies, congenital disorders, immunodeficiencies, and also the immunoglobinopathies. So these are the different types of hematopoietic stem cells that you can find from the bone, from the umbilical cord, and also from the peripheral blood. So what is a stem cell timeline to talk about how all this happened? From when are we talking about the stem cells? So if you can see in this stem cell timeline, 1954, that is so many years earlier, 1954, discovery of murine teratocarcinomas were found out. So they were taken out. The teratocarcinomas are the cells that can divide infinitely. So that is where they found out what is the importance of the stem cells. So discovery in 1954, and also the discovery of human carcinoma cell lines in 1977, and so on and so forth. We came up till 2006, where induced pluripotent stem cells developed. If you remember, I was talking about this adult stem cells, wherein we were talking about the induced pluripotent stem cells being developed. And when you're talking about stem cells, this is the hot topic, where induced pluripotent stem cells is the one that is being used in this particular stem cell therapies. So this was all about stem cells. And hope you have understood what is adult stem cells, what is embryonic stem cells, and what is hematopoietic stem cells. Now we'll go to our next chapter, that is Unit 5. If you can check out in your textbooks, Unit 5, we are talking about propagation and maintenance of tissue cultures is what we'll be talking about right now. And how you're going to propagate, how you're going to maintain the tissue culture. Now that we were talking about stem cells, now we'll go into how are we able to isolate the tissue. How are we able to culture them in vitro, that is outside the cell, and how are we going to maintain those tissue cultures? Is all about propagation and maintenance of tissue culture. So if you can see over there, there's a small piece of tissue in the picture on a Petri plate, and how we are going to take out all these, and how are we going to do this particular chapter. So I hope you have understood hematopoietic stem cells till now, students.
Yes, madam.
Yes, ma'am. Right. So we'll talk about the next topic. This is the tissue culture. So before going into tissue culture, what is cell culture? So what is all about cell culture, and how are we going to propagate them and maintain them? So cell culture refers to removal of the cells from an animal or a plant. So if you can see over there, you can talk about the animal, you can talk about the humans, or you can also talk about the plant. So removal of a tissue and their subsequent growth in a favorable artificial environment. What is artificial environment? In vitro environment. That is, you're going to take it out from its natural cells, and then you're going to grow them outside the cell, outside the environment. It's called artificial environment. And the cells may be removed from the tissue directly and disaggregated. That is, you're going to shear them up by enzymatic or mechanical means before cultivation. Because if you have seen, we're going to take out a tissue. The tissue is going to be a clump. You're going to enzymatically disaggregate, or directly you can also disaggregate them, and then you're going to grow them in the artificial environment. And this may be derived from a cell line or a cell strain that has already been established. What is cell line and cell strain? We'll be talking about it in a very short class of here. So here, if you can see, we have taken a tissue. So this is a tissue over there in a Petri plate, and then we are going to add trypsin. That is, trypsin is enzymatic disaggregation of the tissue over there. So once that trypsin is added to the tissue, the trypsin is going to disaggregate the tissue, and they're going to spread them or separate them. So if you can see over here in the next picture, the tissue is taken. The tissue is mechanically or enzymatically disaggregated. So a solid tissue is where now you're going to find them as single cells, separated cells. These are going to be transferred to a growth medium, and then they are going to be transferred when they grow. They're going to be separated as continuous cell lines or a finite cell lines. That's called primary cell culture. So what's a primary cell culture? Removal of the tissue, mechanically disaggregating them or enzymatically disaggregating them, and then culturing them in an artificial medium. So what is this primary culture? So if you are going to talk about the culture, you're going to talk about primary culture, secondary culture, tertiary culture, and so on and so forth. What is this primary culture? So it refers to the stage of culture after the cells are isolated from the tissue, proliferated under an appropriate condition until they occupy all the surface area available to reach that confluence. What is confluence? Complete growth. If you can see in this picture. So first time when you're going to grow the cells in vitro, you can see a 20% growth. Then later on, after a few hours, you find 50% confluence. Then they're going to grow up till 80% and 100%. So what is 100% confluence over here? That is from 20%, you find that the cells have been dividing and they have been growing extensively. That's called as cell confluence. So whenever you hear the word confluence, remember that they are growing in number. So at this stage, the cells have to be subcultured or called as passage. You have to remember this word also, passage. Passage is subculturing. So, for example, when you're talking about subculturing or passaging, when you're going to make curd, you first take a small bit of, little bit of curd, that is a spoon of curd, and you're going to add it in a milk, a bowl of milk. After some days, it's going to become completely, after a few hours, they're going to turn into curd. Now, when you're going to completely turn into, when the whole milk has turned into a curd, you want to do another vessel because you can't pour the milk into that same bowl because it's going to turn sour. So what you're going to do, from there, you're going to take 1 spoon and then you're going to freshly prepare another set of curd. So that movement, that is from taking a spoon from the formed curd into another bowl and then from there into another bowl, that's called as passage or subculturing. So remember what is passage. So at this stage, the cells, that is the moment it is going to turn into 80%, you're not going to allow them to go to 100% growth of the cells over there, because once they reach 100%, you cannot retrieve them back because there is no more food available and sometimes they might die. So once it has reached up to 60, 70 or 80%, then you're going to remove from this 80% and you're going to do a subculturing or transferring into a new vessel for a fresh growth. So that is called as primary culture, that is being subculture. So from primary culture, when you're going to remove some and removing it and putting it into another fresh growth medium, that's called as secondary culture. That secondary culture becomes a cell line. So that is the cell line that is going to being formed. So what exactly is a cell line? So you have remembered primary culture, removing of the tissue, segregating them or disaggregating them, and then you're going to grow them. That's primary culture. Once that primary culture have grown in amount, you're going to remove that and you're going to subculture them. That becomes a secondary culture, or it can also be called as primary cell line. Cell culture, that's going to cell line. Remember those terminologies over there. So after the subculture, the primary culture becomes known as cell line or subclone, because the same cells you're going to multiply them. That's called cloning. The cell lines derived from the primary cultures have a limited lifespan. That is, they are going to be finite cells. They cannot grow continuously for generations and generations. So they have a limited lifeline. That's called as finite lifeline. And as they are passaged, cells with the highest growth capacity predominate, resulting in a degree of genotypic and phenotypic uniformity in the population. That's why you call this clone, subclones, because they are going to be the same as the parent cell, genotypically as well as the phenotypic uniformity. So that's cell line. What is cell strain? If a subpopulation of a cell line is positively selected from the culture by cloning or some other method, this cell line becomes cell strain. Cell strain is that same strain of that particular parent cell line. It's called cell strain. So a cell strain often acquires additional genetic changes subsequent to the initiation of the parent line. So a little bit of genetic changes you will find that, because as they are copying, that is, as the mitosis is occurring, sometimes there can be some bit of mutations occurring. So a little bit of genetic change will definitely be seen in a cell strain. So if you can see in this picture, from the tissue, you're going to culture them. That's the primary culture. From the primary culture, when you're going to again subculture them into a secondary culture, that becomes a cell line. And from this cell line, when you're going to remove some subpopulation, which are going to be same, and you're going to grow them separately, then that becomes a cell strain. Why a cell strain? If you're interested in one particular cell strain, then you're going to identify them and you're going to remove them and then you're going to culture them. That becomes a cell strain. So here,
Ma'am,
yeah,
ma'am, this cell strain you have told, no, just now.
Yeah.
That once ante ippudu manam oka when we are culturing, we are taking out, as you tell, that pure that perfect something you have said, అది మనం అంటే ఇప్పుడు తోడేసినట్టు కొంచెం తీసి మనం వేరే దాంట్లో వేస్తాం కదా, అప్పుడు గ్రో అవుతాయి కదా అందులో. అంటే దాన్ని సెల్ స్ట్రెయిన్ అంటాం. అంటే ఇక్కడ నుంచి తీసుకొని ప్రైమరీ దాంట్లో నుంచి తీసుకొని సెకండరీ దాంట్లో తీసుకెళ్ళడం సెల్ స్ట్రెయిన్.
Exactly. Yeah. Can you see this picture over there? Can you see this picture?
Yes, ma'am. Yes, ma'am. Not clear. So, little up.
Pardon, ma?
Little. Okay. Now I am able to see.
Yeah. So if you can see in this picture, you have taken a tissue. దాన్ని మనం కల్చర్ చేస్తున్నాం. ఇది ప్రైమరీ కల్చర్ అయిపోతుంది. ఓకేనా?
ఎస్ మ్యామ్.
సో ఈ ప్రైమరీ కల్చర్ కన్ఫ్లుయెన్స్ వచ్చేటప్పటికి కొంచెం తీసుకొని దీని నుంచి తీసి మనం సపరేట్ చేస్తున్నాం into a fresh growth media. Now, when you're culturing them in a fresh growth media, this becomes cell line.
Okay.
Okay, na? So this can be called as secondary culture or primary cell line. Just make a note of it.
Okay.
Okay, na? From primary culture, you're going to take it. So if you can see in this picture. So primary culture nunchi తీసి మనము secondary culture చేసేటప్పుడు ఈ secondary culture becomes
cell line అంటాము.
Cell line, or it is also called as primary cell line. Just cell line అనొచ్చు, లేకపోతే primary cell line అని కూడా అనొచ్చు. సో ఈ ప్రైమరీ సెల్ లైన్ నుంచి మనము సెలెక్షన్ అంటే if you are needing one particular cell of your interest, you are going to identify by using that surface markers. Remember I was talking about CD34 markers. So when you're going to use some markers and you're going to select one type of a cell strain that you are talking about, if you're going to remove that and you're going to culture them separately, then that becomes cell strain.
Okay. Now I get it.
If you're not interested in cell strain and if you're just going to passage this again and again, then you're going to talk about secondary culture, tertiary culture, quaternary, penta, and so on and so forth. That becomes a chain. Got the point now?
Okay. Yes, ma'am. Now I got.
Right. So here, when you're going to talk about the question, they will ask you all, what is the propagation method and maintenance of cell culture? This is what you're going to talk about. You're first going to talk about the primary cultures, and you're going to talk about aseptic techniques. That is, for culturing, what are the necessary equipments that you need, and what are the things that you're going to maintain to have a perfect culture without any contaminations, is what you're going to talk about in this coming slides over there. So aseptic techniques. This aseptic techniques, 1 or 2 slides, I'm going to give you all extra information which is not found in your book. This can be taken for your own interest. Otherwise, you don't have to answer all this in your examination point of view. So when you're talking about biosafety levels, this biosafety levels can be of 4 levels, that is from level 1 to level 4. So biosafety level 1, what is this level over here? Basic level of protection common to most research and clinical laboratories. That is, when you're going to talk about a simple diagnostic center, this is called as basic level, where you give your blood samples for diagnosis purpose, then that becomes a basic level. So that is biosafety level 1. The simple common protection level of protection is found in this particular laboratories. And this is appropriate for agents that are known, not known to cause disease in normal, healthy humans. So handling a blood, you can have normal protection with a gloves, and then you can do this experiment. So that is called as not known to cause disease in normal and healthy humans. So when you're talking about the second level, appropriate to moderate risk agents known to cause human disease of varying severity by ingestion or through inhalation can also cause these diseases. So you're going to take heavy precautionary measures. That comes under biosafety level 2. Most cells culture lab should be at least biosafety level 2. Culture labs, that is when you're going to talk about research purposes, then that becomes a culture lab. There you need biosafety level 2. But the exact requirement depends on the cell lines used and the type of work that is being conducted. So if you can see in this picture over here, biosafety level 1, wherein you have a small room, you have a working desk over there, and then you have an incubators over there that's present, and the person is wearing a common apron and the gloves and proper glasses being used. They have a proper washing area over there, and the soaps and the precautionary measure being kept over there. So when you're talking about biosafety level 2, a little bit of increase, that is, you have a laminar flow. Remember the laminar flow that we were talking about in the first year? So they have a laminar flow. Why a laminar flow? Because you're going to work on a cell strain or a cell line. Then you're not supposed to expose yourself to that particular cell line. So you need to work under a laboratory other than the basic table and an incubator. So when you're talking about level 3, with a little bit of risk-based enhancement. So risk-based enhancement is when you're going to talk about some viruses wherein you're sure that this is going to affect, when you're going to talk about some parasites which is going to be affecting us, then you have to have a biosafety level 3, wherein the person, if you can see, the person who's going to work, he's been properly covered from the head to the toe. He has a mask over there with the oxygen cylinder attached, because he's not supposed to inhale the air over there. And if you can see in this picture, you have a double door room over there, not a single door room like this. It has a double door room, wherein the person who's working over here, this particular fungus or virus or bacteria cannot escape this particular room over there. So you have the laminar airflow, the working setup over there, and a double door room with a safety mechanism. That is level 3 over there, airtight disinfectants. So when you're talking about the biosafety level 4, can you see the persons? They are completely covered from head to toe, as though they are astronauts over there. So completely covered. This is how proper precautionary measures is taken, and you can see even while viewing it under a microscope, they have to be covered from the head to toe. The person working over here on the laminar airflow, he's also completely covered, because they are working under some exotic agents that can pose a high individual risk of life-threatening disease by infectious aerosols, and for which sorry, and for which no treatment is available. So, say for example, when we were working on this, when researchers were working on COVID viruses over there, they didn't know what exactly the procedure is going to be, what exactly the pathogen is going to affect on them. So neither did we have any treatment, although they were doing some treatments, but just normal treatment which they thought would help them. So this researchers, they had to cover from the head to toe and then work. So something like that, this particular biosafety level 4 is needed. And these agents are restricted to high containment laboratories. So this particular room cannot be entered by anyone inside. It has to be a restricted area. So when you're working in a laboratory, what are the safety rules? So here, this point you can write in the examination point of view. So only an authorized person has to enter the laboratory, lab coats to be worn, sensible footwears to be worn, because you don't want to expose your legs either to the particular diseases. Then a caution has to be kept outside the door. And also whenever it is cleaned up, you have to place this cautions to be careful. No smoking, no drinking, no eating. All work must be carried out in accordance with the risk assessment, because you have a protocol, and all those protocol has to be taken care of. Dispose waste material properly, because you can't take a chance. So everything has to be disposed properly in a container over there. And another main important thing is you have to keep your lab neat and clean, and keep everything in handy so that whatever you want, you can be easily accessible to. So you can take it very easily. So if you can see the person working in the laminar airflow, how clean the equipments are arranged. If you can see over here also, everything is arranged neatly so that all the work can be done very easily. So that's a safe laboratory practice. So when you're going to talk about laboratory cultures, you're going to also talk about a little bit about the cell culture hood, laminar airflows. There are three different types of classes of laminar hood over there: class 1, class 2, and class 3. Don't get confused with the biosafety level 1, 2, 3, and 4. Here we are going to talk about the cell culture hood, how safe they are when you're going to talk about its cultures. So here, when you're talking about the class 1, 2, and 3, they are developed to meet varying research and clinical needs. So what are those varying research clinical needs? So the first picture over here, you can see class 1 type of laminar hood. They are significant, they offer significant level of protection to laboratory persons when used in good microbiological techniques, but they do not provide culture protection from contamination because they are not much equipped with the HEPA filters over there. So they are similar in design and airflow characteristics to the chemical fume hood. So it's just equivalent to chemical fume hood. So when you're talking about class 2 over there, this is the laminar hood we were talking about. If you remember, I was showing you all some of the laminar hood which we had in our lab in the class over there. So here, this is in equation with the basic level 2 and 3 materials over here, because they are going to provide an aseptic environment, completely aseptic, that is 99.9% aseptic environment. And the cabinet should be used for handling potentially hazardous material. Like if you're going to talk about primate derived culture, primate is the different types of primates that if you're going to take out and culture them, virally infected cultures, radioisotopes, and carcinogenic or toxic reagents. If you're going to work on all these type of things, then you're going to use a laminar hood, which is of a class 2 type of a laminar hood. So you can see over there, the air that is moving, so this can be captured at some places over there, and through the lower point of the cabinet, the air is passing through, and then they are going to be filtered from the HEPA filters over here, and whatever the air that is flowing inside the laminar air hood is a very clean air that is going to be coming inside. So this is a culture hood of class 2 type. Now, when you're talking about the class 3 type, remember when we were going to give this samples for coronavirus, you must have seen the person, he's putting his hand into this particular hoods over there, the glove coats over there, if you can see in this point, and they from there they are trying to take out the samples and doing that. Because why? As I told you all, this is highly risky type of work they are doing. Because it can cause any type of diseases, and there is no preventive measures for that. So that is the reason why they'll be using this class 3 biosafety cabinet, which has a gas. These are all gas type. If you can see in this picture, there is no opening like what you have over here. There's an opening over here to work. You put your hand inside and work directly. But here, you cannot open the glass doors over there. They're all gas type, completely tightened, and only your hands move inside. Even the hands that are going to go inside, you have gloves over there, so nothing is exposed inside over there. So only the hand with the gloves, you're going to work inside. And also, if you can see in this picture, there is an inlet for HEPA, which is kept over there. And there is a pass box over there. Pass box is if you want to keep some instruments or equipments inside this hood, you are going to open this door, and then there is one more door over there, and then you're going to keep all your glasswares or whatever instrument you want to put it inside. So you are not going to directly open it because these are all airtight closed. Then you have this inlet for air, that is a HEPA filters goes inside, the air goes inside, filtered from here, and then whatever is present inside, Again, you have an HEPA filter over there. Why? Because you're not going to expose the microorganism outside the air. So again, there are some HEPA filters on top, which is going to filter that air, and only the fresh air comes outside. So this continuously goes on. And this is the workplace wherein you're going to insert your hands and then you're going to work about. So that is a class 3 biosafety cabinets, which is a gas tight cabinets over there. So you have two inlets over there. One inlet is for HEPA, that is the air to move inside, and another inlet is for passing your glassware that has to be kept, which is covered by double doors over there. So it is not easy to open and do the work over here. Okay, so why so much of safety has to be taken is because you don't want any biological contaminations to occur in the cell culture. Because when you're doing the cell cultures, you are working so that you're doing, either you're going to screen some drugs over there, or either you want to talk about physiological characters of that particular cell, or you're going to use it for any therapies, or you're going to grow some tissues or organs from that. You don't want any contaminations to occur. So what are the two different main contamination that can occur to your cell structures or cell cultures? So chemical contamination is the first one. This can be due to impurities that you find in the media or the sera that you're using and the water. And of course, there are some endotoxins, the plasticizers, and the detergents, because detergents is when you're going to wash the vessels, the different types of glassware that you're going to use. This can cause chemical contamination. So when you're going to talk about a biological contamination, it can be caused due to bacteria, mold, yeast, virus, mycoplasma, as well as cross-contamination by other cell lines, probably. And by following good aseptic techniques, this can be eliminated. So what are the type of biological contaminations? When you're talking about the biological contamination, when you're going to view your culture, you can see some growth that are present over there. So if you can see, so this is possibly the culture that has been affected by mycoplasma colony. What is a mycoplasma? These are very minute type of bacteria. These are also one type of bacteria. They're very minute, microscopic, self-replicating type of bacteria over there. And there are some cell debris that are found. So this is completely contaminated. And when you're going to talk about another cell culture over here, there is some small patch over there. And when you're going to talk about this, enlarge the small patch, you can see how many E. coli growth can be found. So this is again some bacterial infection in the culture. And if you can see the next one, you can also find some yeast growth that is grown in the cells. This is again one more type of contamination. And this is how you can see the fungal mycelium, if you can see over here, enlarged one over there, the fungal mycelium. So these are the different types of contamination that you can find in the cultures if you're not going to maintain proper aseptic condition. So morphology of the cells in the culture. So what is a morphology? That is, morphology is you're going to even at the same time when you're going to check for the contamination, you're also going to check for the shape of the cells under the cultures over there. So what are the shapes over there? The cells in the culture can be divided into three basic categories based on the shape and appearance. That is morphology. So if you can see in this picture, we are going to talk about this fibroblastic or fibroblast-like cells, which has a bipolar condition. Bipolar is two sides, polarity on two sides over there is a bipolar, or sometimes it can also have multipolar. If you can see in the cell over there, they have a multipolarity over there, there and there. So this, and bipolar is two sides over there. This is one side and that's the one side. So bipolar or multipolar, they have elongated shape and grow attached to the substratum. So that is, when you're going to grow them in a culture plate, they're going to get attached to the substratum and they're not going to be as a suspension. Now, this is a microscope that I've kept. This is an inverted microscope. And if you can see the plate that I've kept over there, the plate is kept on the working plate where you can view them. And these are the lens that is kept in underneath the plate. That is why it is called as inverted microscope. And whatever you can see from this eyepiece to check what is the shape. And this is where the light is kept on them. Remember the common simple microscope, you have the light beneath and you're going to view them underneath. But inverted, is the lens is below. You're going to place your plate, and then you're going to throw some light on from the top, and then you can see whatever the cells that are present there. So fibroblast, this is the proper shape that you find in a fibroblast. So when you're talking about the epithelial like cells, they are polygonal in shape. If you can see in the picture over there, they are polygonal in shape. And if you're going to talk about the lymphoblast, mostly spherical in shape is what you're going to see in them. And these are the culture plates or culture instruments that you'll be using. These are the flasks, these are the petri plates, these are the plates, the 6-well plate or 12-well plates or the 80-well plates, different types of plates that you can find to grow them. So these are the equipments or you'll be using for the culturing purpose. Okay, so how you're going to isolate? So first and foremost, now that you have understood the aseptic techniques and how you're going to safeguard them from any contamination, the next thing comes is what and how you are going to isolate the tissue from where and which animal. So when you're going to isolate, you cannot do it on any type of animals because you need a permission. And that permission, you have to get it from the CPCSEA. What is CPCSEA? That is a committee for the purpose of control and supervision of experiments on animal guidelines. These people are very, very strict when it comes to working on animals, because you cannot use the animal how and whatever wish you can do it. There is also one more, along with the CPCSEA, there is one more institutional animal ethic committee. Institutionals, if you're going to work in any university or any institution, they have their own animal ethic committee, and you also have a committee for the purpose. That is people from outside, they come and supervise the experiments that is being conducted on the animals. So you are going to take both these permission from these two ethical committee before taking out or isolating the tissue. So work with human and animal tissue should be carried out in class 2 biosafety cabinet. Now remember, the class 2 biosafety cabinet, you're going to work under them, and dissection should not be done in tissue culture lab to avoid contamination from animals. Okay, so that was how we are going to do the isolation. Now we are going to talk about from which animals and what is the date from when you can take out these tissues. So when you're talking about the isolation of mouse embryo, here remember we are going to talk about mouse embryo. The mouse embryo, you can clearly take it from the day 13 to day 14, that is after fertilization. Stage 2, the day 1 is a 2-cell stage, day 1.5 is 4-cell stage, and so on and so forth. So on day 13 or 14 is a time for the embryo to be isolated. So if you can check over there, this is a picture how you're going to isolate the embryo. So you're going to make a small incision at the abdominal end of the mouse, and after taking out the incision, you can clearly see the embryos over there, the small embryos over there. You're going to squeeze out the skin outside over there, and you're going to isolate the embryo. You're going to place it on the petri plate, and these are different. So you have 1, 2, 3, 4, 5, 6, 7, 8 embryos over there, and you can clearly take out one small pieces. You're going to squeeze it out and see you can remove one embryo like that. So that is how you're going to do a mouse dissection for taking out the mouse embryo. In a colored picture, if you want to check it out. So here you're going to first swab it neatly with the alcohol 70% on the abdominal end. Then you're going to cut open the skin, and you can clearly see the embryos over there. You're going to take out the embryo, utri filled with the embryos over there, and you're going to place it on the petri plate. And this is a even more closer shot of the point over there. So this is the placenta and the uterus over there, and this is how you're going to take out with the forceps. So if you're going to take it out, so tweezing should be done very carefully, and once tweezing is done, you can see the different the number of embryos that has been taken out and placed in the petri plate. So you have this many numbers of embryo of the mouse. Okay, so when you're talking about the chick embryo, so if you remember, we had done this isolation of the blastoderm and the chick embryo. If you remember, in the first year, we were talking about the 24 hours, 48 hours, 72 hours. So when you're talking about the chick embryo, when you can remove them, it is best taken out on the 8th day of the incubation period of the eggs. So on the 8th day, you can easily isolate the embryo. That is, again, you're going to swab the egg with 70% alcohol, and you're going to place the blunt forceps facing up, and then blunt area facing up, and you're going to just slowly crack open the area over there, and you're going to take it out with the forceps over there. So this is how you're going to peel it out and take it out with the forceps. So gently you're going to take it out there. So you're going to place them on the Petri plate, and this is when you're going to view it under the microscope, you can see that large eyes over there, the brain over there, the forebrain, the midbrain, and the hindbrain. That's very clearly see them. So this is even more a clear picture of what you're going to view it over there. So that's the forebrain, the midbrain, and the hindbrain, the eyes with the lens thoroughly seen, and you can also see the heart over there, which can be clearly seen function. So when you take out this brain and place it, this is how it is going to look like, with the eyes over there, and the forebrain, the midbrain, and the hindbrain. Then not only the brain part, you can also take out the rudiments of the chick embryo. So what are the rudiments? If you want to get a good reproducible culture, you can take it out from the day 10th to 13th day of the incubated chick embryos, and you can easily dissect out the individual organs or the tissues and place them preferably in a cold trypsin overnight. So what you're going to do first, you're going to take out the embryo, that is the 13th day embryo. You can see over there, this has been isolated. On a 13th day embryo, you can see the wings that have the forelimbs or the wing buds, which has slowly increased in size, and this is the legs, future legs, which has increased in size, the tail region and the head region over there. So what you're going to do, you can just cut out the head region over there, and then what you're going to do is, if you're going to work on a retina, you can remove the eyes, and then you can peel off the retina and scooping out of the brain is also done over there in the F part. So this is how you're going to scoop out the brain. This is how you're going to take out the retina over there. And also you can also take out the heart, the lungs from the anterior half of the chick embryo. And from the posterior half, you can remove the kidneys and also the different, the thigh muscles from the thigh region of the chick embryo. And you're going to place them clearly over there. So what are the things that you can find over there? You can find the brain, the heart, the lungs, the liver, the gizzard, the kidney. Then also you have the spinal cord over there, the skin, and also the muscles. So why, you must be wondering, why are we taking out all these small pieces and keeping it over here? Remember, we are doing stem cell cultures. So culturing of the different tissue, you're going to take out this particular tissue and you're going to culture them to see what are the type of the cells, the morphology of the cells, and if you're going to do some therapeutic work, how the drugs are going to work on different cells is what we are going to talk about. And so for that, basically you want a tissue. That tissue, how you're going to take it out is what we are going to, we have just now talked about. So preparation of the primary culture over there. So several techniques have been devised for disaggregation. Disaggregation is, now that you have removed the tissue, you cannot use the tissue directly for the culture. You want to separate the cells. So that separation of the cells from the tissue is what is called as disaggregation of the tissue, isolated from the primary culture. So when the isolated tissue is placed in a suitable culture, they will attach, divide, and grow, and this is called as primary culture. So what is the technique used? This can be divided into enzymatic disaggregation, that is, some tissue do not require enzymatic disaggregation. So those tissue are the soft tissues which do not need enzymatic disaggregation. You can directly use them for culturing. Then the second one is utilizing the enzymatic disaggregation, and this enzymatic disaggregation gives a better yield when more tissue is available. That is, when you have enough tissue to disaggregate them, then you can use this. And third one is purely mechanical technique. Mechanical technique involves how you are going to do it with your own hands, the maceration of the tissue, which works well in a soft tissue, because soft tissues are very, very soft. That is why the name soft. And when you're going to add enzymes to them, that might kill the cells, or you might lose some important informations on that particular cell. So for them, mechanical technique is used. So what are all these? We are going to talk about them. So if you can see this chart over here, isolation of tissue is the first one. How you're going to isolate it? We have dissected it and removed them. And once you have dissected it, you're going to undergo 3 different types of disaggregation method. One is fine dissection, that is chopping down to the explant size, that is very small size. Then the next one is a mechanical disaggregation, that is by sieving, that is sieving them, syringing them, or vigorous pipetting them. And the third one is a enzymatic disaggregation. Enzymatic disaggregation, will be talking about cold trypsin, warm trypsin, and another type of enzyme called the collagenase over there. So first we'll talk about fine dissection, that is which do not need any enzymatic disaggregation and culturing them. So what is the protocol? You're going to remove the embryo from the eggs and place them. See here, the protocol is for the embryo from the eggs. It can be also used for embryo of the mouse also. So here I'm just talking about the egg over here. You're going to place them in DBSS, the buffered saline, the dissection buffered salines over there. So you're going to remove the head, remove the eye, and open it carefully, dissect and gently peel the pigmented retina, expose the head. So this is what we have talked about. After the exposing and removing all these parts, you're going to transfer all these tissue to a separate test tube containing 1 ml of 0.25% trypsin, and you're going to place them on an ice. Leave them over there for 6 to 18 hours at 4 degrees and carefully remove the trypsin from the test tube without disturbing the tissue. So here you can see, you're going to take out the tissue, you're going to add it in the PBS, phosphate buffered saline or dissection buffered saline. Then you're going to add 0.2% of trypsin and 0.01% of EDTA. And then you're going to leave it overnight, incubate it, leave it overnight, and then or sometimes at 15 to 20 minutes at 37 degrees. Then you're going to remove the trypsin, you're going to add the medium, and then you're going to culture them directly without doing any much work over there. So that is first part of dissection over there. So this is what I was talking about in this picture over there. So you can just go through this protocol or the procedures. And this flask is a 25 centimeter square flask. So this is a 25 centimeter square flask is what you'll be using for culturing different types of tissues over there. So the next one is a mechanical disaggregation. How you're going to do? By mechanically. So what is mechanical disaggregation? One is by scraping. That is with the help of the knife, you're going to scrape the tissue and you're going to make them into small, small pieces. The second one is sieving. Sieving is how you're going to sieve your atta and all those, something like that. Here you're going to use very small type of sieving mechanism over there. This is another type of mechanical disaggregation. The third one is a syringe. With the syringe, you're going to push it up and down so that you're going to break them into smaller pieces. And of course, pipette type of this thing where you're going to remove and take it and remove out the tissue so that you're going to vigorously pipetting, and hence you're going to break the tissue into pieces. So that is mechanical disaggregation. So here, if you can see in this picture, this is what I was talking about, sieving mechanism. This is a centrifuge tube. We all know centrifuge tube over here. They are attached to a small sieve over there. So that is a sieve over there, very small micron sieves over there. And this is a rod, wherein this rod is used to, when you keep your tissue over there, then you're going to press it with this particular rod so that the paste, whatever the paste is coming out, it can be collected into the test tube. So that is one type of mechanical disaggregation over the test tube or a centrifuge tubes or a Petri plate dish that can be used over there. So the mechanical disaggregation method involves collecting the cells when the tissue is carefully sliced, pressing the dissected tissue through a series of sieves for which the mesh is gradually reduced in size, and alternatively forcing the tissue fragments through the syringe, or simply pipetting it repeatedly. So here you can see, so this procedure gives a cell suspension more quickly than does the enzymatic digestion. You're just going to press them and you're going to get the tissue. So this is a little bit faster. We're going to talk about enzymatic. We'll talk about enzymatic digestion here. Scraping and sieving are probably the gentlest mechanical method, while pipetting and particularly syringing are most likely to generate shear. That is, when you're pipetting or when you're using the syringe, a little bit of shear or wear and tear of the cells can occur. And this is a process preferred when working with bone marrow, lymph nodes, spleen, and other tissue that are not bound too tightly. Remember, when you're talking about mechanical disaggregation, we're talking about soft tissue. So when you're talking about soft tissue, the bone marrow is soft, lymph nodes, spleen, and other tissues which are very soft, they can be used for mechanical disaggregation. So when the availability of the tissue is not a limitation and the efficiency of the yield is unimportant, mechanical disaggregation can be followed. So bone marrow, you will have enough, lymph nodes, you have enough, spleen also you have enough. So when you think that a little bit of wear and tear is no problem, then you can go for this mechanical disaggregation. So here you can see over here, you have a centrifuge tube over there, and you have this lids over there. This particular lid is enlarged over there. If you can see, you're going to add the tissue onto this lid, and then you're going to add a little bit of buffer, and you're going to cover them with the sieve. And what you're going to do is you're going to keep rotating them so that you're going to make them into paste. Remember, when you take an orange, you want to take juice out of it. So you have those juice, mechanical juice. I'm not talking about the mixie, but mechanically, manually, you're going to take it out the juice. So something like that, you're going to work in this particular tissue dissociation there, and you easily can collect them into the centrifuge tubes. So this is the pictures. You can see the protocol over there. So what is the protocol? He says, after washing and dissecting the tissue, it is chopped into small pieces of 3 to 5 mm and placed into a stainless steel or a polypropylene sieve. So these are the polypropylene sieves that you find over there of 1 mm mesh and a 9 centimeter Petri dish, or also you can keep them in a centrifuge tubes. The tissue is forced through a mesh into the medium by applying a gentle pressure with a piston or disposable plastic syringes. So you're going to place the tissue over there and you're going to gently press them so that everything is getting collected into the centrifuge tubes. And the partially disaggregated tissue is pipetted from the Petri dish into a sieve for finer porosity. So you have this different types of porosities over there. Whatever type of porosity you want, you can just select them and you can thoroughly disaggregate them. The culture flasks are seeded at 2 into 10 to the power of 5 or 1 into 10 to the power of 6 or 2 into 10 to the power of 6 cells per ml. So 1 ml contains this many number of cells that you have been going to add into this media over here. So diluting the cell suspensions in the media. So this is a flask containing the cells and the media. The pink color one is a media over there. So that was about mechanical disaggregation. Now you're going to talk about enzymatic disaggregation. So enzymatic disaggregation needs cold trypsin, warm trypsin, and collagenase. We're going to talk about 3 of them. So first one is a enzymatic. First, you understand what is enzymatic dissociation or disaggregation. Enzymatic dissociation uses specific proteins to disaggregate cells, cell culture sample. So the process applies enzymes like trypsin or collagenase. So mostly you will use these 2 type of enzyme, trypsin and collagenase, that digest the pieces of tissue to release the target cells. Enzymatic dissociation is used for more compact tissues, which may have a large quantity of debris. Compact tissues where, for example, when you're talking about the muscle tissue, it's going to be very compact, very thick. You cannot tear them very easily. Okay, so that is compact tissue. And they are also going to have large amount of debris. You have those collagen, for example, you have those ligaments attached, the collagens, the cartilages attached. So you need to use this type of enzymatic dissociation. Using enzymes can reduce the amount of fibrous connective tissue, resulting in a higher yield of cells from the organs such as liver also. So if you can remember, the liver also has a lot of debris around them. Okay, so coming to cold trypsin disaggregation. So cold trypsin method usually results in a higher yield of viable cells with an improved survival of cells after 24 hours of incubation. So after chopping and washing, you're going to keep the tissue in a while and soaked with cold trypsin for about 6 to 24 hours. That is cold trypsin that was kept in the fridge prior. The trypsin is, however, removed and discarded, and the tissue pieces contain residual trypsin. Even after removing the trypsin, it also has a little bit of residual trypsin left over. So these tissue pieces in a medium are incubated at 37 degrees for 20 to 30 minutes. Then the cells are dispersed by repeated pipetting, and the dissociated cells can be counted, appropriately diluted, and used. So this major limitation is it is not suitable for disaggregation of cells from large quantities of tissue. That is, if it is going to be very large, then you cannot use this type of cold trypsin disaggregation. If you can see in this picture, so you have collected the tissue, and then you're going to chop them into small pieces. You're going to wash them, and then you're going to keep them in the cold for some time, and then remove this trypsin. And this is going to have a little bit of trypsin, and that can be dispersed by pipetting n number of times, and then you're going to seed them and you're going to culture them. And this is, you're going to check them for viability, that is whether the cells are alive or not. Now, the next one is warm trypsin disaggregation. What is warm trypsin? Opposite of course, the cold trypsin over there. So in the warm trypsin, the chopped tissue is washed. So here you can see the chopped tissue is washed with DBSS. What is DBSS? I told you, dissection balanced salt solution, or phosphate buffered saline, something on the same line it is. So the contents are stirred at an interval of every 30 minutes. The supernatant containing the cell can be collected. What is this magnetic stirrer? So here, when you're going to talk about this warm trypsin, you're going to warm the trypsin. Along with that, you're going to keep stirring them. That is, after you chop the tissue, you're going to wash by resuspension in the trypsin solution. Then you're going to add it to a stirrer over here. The stirrer, it's going to be stirring the chopped pieces in a warm trypsin over there. And as it is stirring, the supernatant is collected time and again, and then it is added into another vial over there. Then you're going to pull it, and then you're going to view it under the microscope whether it is viable or not, and then you're going to culture them. So again, we'll come back to this. What is it? After removal of the trypsin, the cells are dispersed in a suitable medium and preserved in the ice. The process of addition of the fresh trypsin and incubation and collection of dissociated cell is carried out for about 4 hours. The disaggregated cells are pooled, counted, and appropriately diluted and incubated. So if you can see over here, this is different types of flask over there. That this flask is attached to a stirrer, magnetic stirrer. Once you have collected the tissue and washed it, you're going to add it to a warm trypsin over there. And this magnetic stirrer is going to continuously stir the solution. As it is continuously stirring the solution, you're going to collect after every 30 minutes. You're going to remove it from the stirrer, leave it for some time. All the pellets are going to get collected underneath the heavy tissues that have not been dissociated or disaggregated properly. They're going to settle down at the bottom of the flask. The supernatant is collected every 30 minutes, and this continuous process of stirring and removal is done for 4 hours continuously until you have made sure that you have dissociated the cells thoroughly, and then you're going to go for cell cultures. So the next point is disaggregation by collagenase. So we have talked about cold trypsin, warm trypsin. Now you're going to talk about collagenase. So collagenase is the most abundant structural protein in the higher animals. So you know what is collagen? All those tissue that is having that elasticity has the collagen over there. The collagenase is an enzyme over there. We are going to talk about that. And it is mainly present in the extracellular matrix of the connective tissue and the muscles. The enzyme collagenase can be effectively used for disaggregation of several tissues, even a normal and a malignant one. Malignant is a cancer one, and they may be sensitive to trypsin. So all those cells that are sensitive to trypsin, when you're not going to get a good yield from dissociating them from the trypsin, then you're going to use collagenase for that different types of tissue. So this technique is very simple and effective for many tissues. That is, for example, when you're talking about embryonic, adult, normal, and malignant, and it is of greatest benefit when the tissue is either too fibrous or too sensitive. So tissue disaggregation in collagenase, this is how the outline is over here. So you're going to place the finely chopped tissue in a complete medium containing collagenase and incubate them. And when the tissue is disaggregated, remove the collagenase from the centrifugation, seed them at high concentration, and culture them. So you're going to chop the pieces, you're going to incubate in the media containing collagenase, then you're going to pipette to disperse the tissue, and then you're going to separate them, wash them, and you're going to grow them in different growth medias. That is tissue disaggregation and collagenase. So the protocol you can just go through because I've already talked about what are the protocols over there. That is, after removing the tissues, you're going to transfer these pieces into a 25 centimeter square flask. Centimeter square. This is 2 is superscript over there, and 100 to 200 pieces to a second flask. Drain off the DBS. You're going to add 4.5 ml of growth media and add 0.5 ml of crude collagenase. That is 2000 units per ml is a final concentration, and you're going to incubate them. And after incubation, you're going to culture them, and then you can see the different dispersions over there. This is before dispersion. Cell clusters from human colonic carcinoma after 48 hours of dissociation. And this one is after removal of the collagenase, further disaggregation and pipetting and culturing for 48 hours, you're going to get a very clearly defined round clusters. So this was before collagenase treatment, or just when you've added the collagenase. This is how the cells look like, and this is how the cell looks like after the collagenase treatment, and that is after culturing at 48 hours of cultures. So this is how differentiation you can find in culturing them. Okay, separation of viable from non-viable. What do you mean by separation of viable from non-viable is, viable is the cells that are alive, non-viable is the cells that are dead over there. So what, how we are going to do this removal. So when an adherent primary culture is prepared from dissociated cells, non-viable cells are removed at the first change of medium. That is, when you're going to change the medium. Why you want to change the medium? Because as the cells are growing into confluency, they need more food because they have increased in size, and sometimes also the pH may change. So that is when you have to keep changing the media over there. So with primary cultures maintained in the suspension, non-viable cells are gradually diluted out when cell proliferation starts. Proliferation is when the cell is starting to grow. If necessary, however, non-viable cells may be removed from the primary disaggregation by centrifugation the cells on a mixture of phycol and sodium metrizoate. So you want to remove the non-viable. How you're going to separate the non-viable? By adding the culture media with phycol and sodium metrizoate. So this is example over here is hypic or triosyl is also the name that is given over there. The viable cells collect at the interface between the medium and the phycol metrizoate, and the dead cell form the pellet. So for example, you have a culture over here. The adherent cells are attached onto the culture plate over there. And when you view it under the microscope, this is what the non-viable cells, the dark cells are the non-viable cells, and the light color transparent ones are the viables. Viable is the living cells over there. So how we are going to separate them? You're going to add the phycol and sodium metrizoate into the media over there, and you're going to centrifuge them. After centrifugation, you can see a suspension over there, the phycol media over there, and this is where the viable cells collect at the interface between the media and the phycol metrizoate. So these are the cell. This is the media. This is the viable cell, and that is a phycol. And this is a place where the non-viable, the dead cells are suspended over there. And this is the cells that we are interested in, and this can be collected and they can be again grown in the culture media. So maintenance of culture. So again, you have to maintain the culture. It's something like when you're making curd, you cannot keep on adding the fresh milk into that same bowl over there. You have to remove it, and you are going to culture them in different plates over there. Why? You want to avoid that sourness in that curd over there. The same way, when you're going to culture the cells over there, you want to maintain them. So there will be one more question. describe how you're going to maintain the cell culture is what you're going to talk about in this. So maintenance phase begins when the cells have attached to the surface of the culture dish. So you can see this is a culture dish over there, and the cells have got attached onto the base of the culture dish. Attachment usually occurs 24 hours after initiation of the culture. When the cells have reached a desired percent or cellular confluence, remember what is confluence over there, and are actively proliferating is dividing, it is a time for subculture. So here if you can see, this is a 20 to 30% of confluence, and this is a 50 to 60% of confluence, and this is a 100% confluence. You're not going to allow the cells to become 100% confluence. When is a time for a proper subculture is when you find them 80%. At 50 to 60% of confluence is the best phase when you're going to remove it and subculture them. So that is how you're going to maintain the cultures over there. Propagation of cell lines. Remember what is cell line over there. Now, how you're going to propagate, how you're going to grow them, grow the cell lines. That is, after establishing a cell line, it needs to be propagated in order to characterize and storage for further use. You're going to store them also for further use, because once you have done a cell line, if you're not going to use them, you're going to store them, right? You don't want to waste their culture purpose. Remember, if you're going to do a passage, some cells have very less life, that is finite. Say, for example, if you have subcultured them for 10 times, 50 times, or 100 times, after that, the cells are going to die. So to avoid that, you're going to store some, and some you're going to do the culturing work so that you can do some experiments on them. So once the culture is confluent, it is transferred to the maintenance medium, which provides nutrients to keep the cell alive, healthy, but reduce the replication rate so that the cells do not overgrow. You don't want them to go faster. The content in the flask is very simple to split, and this is done as follows. How? View the culture using an inverted microscope. So you're going to use them. You're going to keep the plate over there. View them under the microscope and assess the degree of confluence and confirm the absence of bacterial and fungal contamination. So if you're going to view them, this particular thing is a fungal mycelia that is grown. Again, contamination out there. And in this picture, you can find the bacteria rods in between the viable cell suspensions over there. Again, this is useless. You're going to throw them out. Until and unless you are sure that there is no contamination, you're going to view it and use only those that are not contaminated. So once you have seen everything is fine, you're going to remove the spent medium, resuspend the cells with a fresh growth medium. What do you mean by spent medium? The medium is void of growth factors. That is, it is void of proteins, carbohydrates, or the minerals over there. So you're going to add them with fresh growth media so that they can divide and they can grow in normal conditions. You're going to record the cell line, clone number, and passage number. So what do you mean by passage number, clone number, and cell line? So this is the first passage, and this passage, when you're going to separate it and going to add it into fresh media, that is, you're going to split them into 1:2. So this is the cell. For example, remember that this has come to a 50 to 60% confluence. So what you're going to do, you're going to separate them into 2 equal flask over there. So you're going to add that fresh amount of growth media, and you're going to separate them, and you're going to grow them again. Now the first passage, when you're separating them into 2 flasks, then this is called a second passage. So passage number 2 is what you're going to name them. The passage number P2 is what you're going to name them. Okay. And subculture of adherent cell line. What do you mean by adherent cell line? So adherent is the one that gets stuck to the base of the culture media. So this is the flask. You can find all the cells attached onto the bottom of the flask over here, that is called adherent. When the cells are not attached onto the surface, the bottom part of the flask, and if you're going to move, then you're going to call them as suspension cell lines. Suspension is they are floating in the media. That's called a suspension cell line. When they're going to attach onto the bottom of the flask, then you're going to call them as adherent cell line. So remember these 2 names, adherent and suspension cell line. So what you're going to do, how you're going to subculture the adherent, because these have got attached onto the cell. So how you're going to remove them and how they're going to split them is what you're going to talk about in this protocol. Assess the culture, remove the spent media, wash the cells with PBS, without calcium and magnesium. Repeat if required. We're going to wash them first because you want to remove the spent media. We're going to wash them. Pipette trypsin EDTA onto the cell monolayer, rotate the flask and decant. You're going to add trypsin. Why trypsin? You want to dissociate them from the cells, from the walls of the flask. So you're going to remove them over there. So that's the reason why you're going to add trypsin EDTA over there. And you're going to rotate the flask very slowly, and then you're going to decant the trypsin again. Incubate for 2 to 10 minutes and examine the cells. If you still find the cells are attached to the wall, you are going to again add the trypsin, repeat that same if necessary. Then you're going to remove it once you're sure that all the cells have been suspended into the media. You're going to add fresh media into that, the serum containing media, to inactivate the trypsin. Transfer the cells into the fresh, warm, new media. Incubate if required or as required, and repeat if necessary. So that is how you're going to do the subculture of adherent cell lines. So how you're going to quantify the cells in the culture? Remember, when we are doing the RBC count and the WBC counts in the first year with the Newburst chamber, this is how we are going to do it under the microscope. A viable cell count is crucial for the study of eukaryotic cells for different purposes, such as management of cell culture in biological research, titration of cell population in diagnostics, and also in the process of control in industrial process. So if you can see in this picture over there very clearly, this is a Newburst chamber, and this is one side of the chamber, which is 4 into 4, that is 16 blocks is what you can find over there. This is the image of keratinocytes in a hemocytometer viewed through a phase contrast. And note that the cell morphology is heterogeneous, different types of there. And there are some bright cells, and there are some light color cells over there. The dark looking cells and the debris should not be counted because these are the dead ones over there. The black color ones, these are the dead ones. The red ones, the light color ones, is what is the viable cells. Those are the cells that you're going to count. So that's how you're going to find out the viable to the non-viable cells. So cell counting can be of two types: direct method and indirect method. Direct method by adding them onto the hemocytometer, or also electronic cell counter. And indirect method is protein and DNA estimation or glucose uptake. So what are these methods? So the direct method is the hemocytometer method, that is the Newburst chamber. And this is a specialized thick glass. If you remember when you're talking about this, you must have done it in your cell and mol bio also in the first year, and in the second year also you must have counted all this in the Newburst chamber. Yes, ma'am. Do you all remember this Newburst chamber? We have done in the practical.
Yes, ma'am. Yes, ma'am.
Yes, madam.
Yeah.
Yes,
Yes,
madam.
ma'am.
RBC counting.
Yeah. So this is a
Yeah.
specialized thick glass over there, and you're going to add your samples in this silver color ones that is placed over there. And when you view it under the microscope, these are the wide ones. So this is divided into 9 large squares: 1, 2, 3, 4, 5, 6, 7, 8, 9. So these are the 9 large chambers that they have, and the cells are counted either in this. So when you're talking about the WBCs, you're going to count in this 4 chambers, and when you're going to talk about RBCs, this is where you're going to count the RBCs over there. Okay, so that was about how you're going to talk about in the Newburst chamber. When you're going to talk about the electronic cell counter, you have this electronic machine that is present over here, because life is made easy in the research also. And so the electronic cell counter is coming to use over there. So what is this? This method involves drawing a suspension of the cell through a small aperture across with an electric current, wherein an electric current is maintained. As the cell passes through the aperture, if you can see over there, as the cell passes through the aperture, there is a counting orifice over there, wherein the electronic detector is going to detect how many number of cells are being passed through, and they're going to count, and how many of our cells is passing through, the number is counted over there automatically. So this is the number of cells that has been counted over there. So this is electronic cell counter machines. Now, when you're going to talk about the indirect cell count, this can have this turbidity. Spectrophotometer is one type. Estimation of cell components, that is a protein, DNA, RNA, and ATP. And online monitoring of carbon dioxide production or oxygen utilization is what you'll be talking about over there. But DNA and protein assay are inaccurate as they do not distinguish between viable and non-viable cells. So this is the turbidimetry method, wherein when the sample is kept over here through the filters, you're going to read out what is the amount, or what is the percentage of cells that is present over there, and under spectrophotometer, what is the OD values that is counted, is what we'll be talking about in indirect method of cell count. So cell viability, how you're going to check whether the cells are viable or not, this can be done by using trypan blue. Trypan blue is a stain that you'll be using. So once you add this trypan blue to the cells and incubate them for a small period of time, when a cell suspension is mixed with this dye, you can see in this picture over there, all the non-viable or the dead cells are going to stain dark in color, because the whole cytoplasm is going to take up the stain and they're going to form dark in color. So that means they're all dead ones. And the viable ones are the one that is transparent in the center. That is, that cytoplasm is not going to take much color. So they are transparent. So these are viable. So that's how you're going to differentiate between a viable and a non-viable by using trypan blue. So the procedure, how you're going to do this procedure, you're going to place 50 microliters of cell suspension in a cryo vial, add equal parts of 0.4% trypan blue to the suspension to obtain a dilution of 1:2. And you're going to incubate the mixture for less than 3 minutes, not more than that. You're going to just leave it only for 3 minutes at room temperature, and you're going to immediately view it under the microscope to count the viable and non-viable. If you're going to take a little more time, say for example, if you're forgotten the time and you're going to view it at 5 minutes interval, then you're not going to get a correct picture over there. So you have to be very careful when maintaining the time. 3 minutes is what your incubation is all about. So yeah, this is again the same thing when you're viewing it on a hematocytometer, how you can see the cells over there is what is given over there. Cell viability using lactate dehydrogenase. This is one more small point that he has asked in your textbook over there. You can also check the cell viability by using lactate dehydrogenase, because lactate dehydrogenase is an oxidoreductase enzyme that catalyzes the interconversion of pyruvate to lactate. So you can go back to biochemistry over there, the biochemical pathways from pyruvate to lactate. Cell releases LDH into bloodstream after tissue damage or red blood cell hemolysis. So whenever there is a hemolysis occurring or whenever there is a tissue damage, your blood content will be increased with LDH. Why? Because cells are going to release LDH into the bloodstream. So since LDH is a fairly stable enzyme, it has been widely used to evaluate the presence of damage and toxicity of the cells and the tissues. So simple method over there. So whenever you have a lot of hemolysis or whenever there's a tissue damage, when you take out the blood and do the biochemical assay for lactate dehydrogenase, if the lactate dehydrogenase amount increases, that means then there is some tissue damage occurring. Of course, there is a little bit of tissue damage occurring every time, but when the amount increases, then there is a time to say, yes, there is some problem in the tissue or in the blood cells. So where all this can be applied, the cell culture is one of the major tools used in cellular and molecular biology, providing excellent model system for studying the normal physiological and biochemistry of the cells, the effects of the drugs and toxic compounds on the cells. And of course, the metabolic studies for aging is also done, the mutagenesis and carcinogenesis. It is also used in drug screening and development and large-scale manufacturing of biological compounds like vaccine and therapeutic proteins, because there are some proteins that are going to be less. So this can be used for screening of that major problems. The major advantage of using cell culture for any of these application is the consistency and reproducibility of the results that can be obtained from using batch of clonal cells. So anytime you do, each time you do, you're going to get that correct reproducible results. So that's the reason why cell cultures are used for different type of any research that you want to do. Okay, so when you're talking about the different types over there, you can also talk about these are used as a model for studying the basic cell biology and biochemistry. This can be used for interaction between cell and disease causing agents, between bacteria and virus, effect of drugs, and process of aging. Cancer research is also one of the most important one. Why? Because we want to bring out some new drugs, and how this cancer can be controlled is again can be done with research cell culture. Because when you have a cell, by adding the drugs that you have been using, you have developed, it will become used very simple, because you cannot use a drug on a human being, which is unethical. So cell cultures can be done very easily, and the normal cells can be converted into cancer cell by using radiation, chemical, and viruses. And the cell culture can be used to determine the effect of drugs for selectively destroying only the cancer cell and not the normal cells. And of course, in the virology, that is development and of viruses, because some viruses are used for DNA engineering purpose. So you can also use them for study and the growth of development. And the mode of infection, what is a different type of mode of infection. Toxicity testing, that is when you have preparing a new drug, what is the effect in the cosmetics and chemical industry also, especially liver and kidney, what is the effect that is going to have, because toxicity means especially it affects the liver and the kidney first. So what is the toxicity level at the liver and the kidney level is also one more important way the culture cells can come into use. Not to forget the vaccine production. Cultured animal cells are used in the production of viruses, which are used for producing vaccines, thanks to the different vaccines that have come up in recently. So examples are polio, rabies, chickenpox, measles, and hepatitis. And also for the COVID, also you have made these vaccines. And genetically engineered proteins. So there are some proteins which cannot be produced by the normal in a disease individual. Then you're going to use this cell culture for protein engineering. That is, you're going to produce commercially important genetically engineered proteins such as monoclonal antibodies for immunology purpose, insulin for diabetes, hormones, and much more can be used. And this can also be used for replacement of the tissue and organ. So nowadays we are coming up with lots of tissue and organ replacement. You can clearly grow them. That is, for example, if you want to do a skin graft, you can take out the skin cells from the person, autography, and you're going to culture them and put back that same skin grafting for that same person, so that there is no sensitivity problem in that particular treatment over there. So this is one way of culturing and having your own cells in your body. So this artificial skin can be produced using this technique to treat people who are suffering from burns and ulcers. And on the same line, you can also culture the liver, kidney, and pancreas, although this is still in a nascent level. Nevertheless, this can also be used. Then fetal cell culture extracted from pregnant women can also be used to study or examine the abnormalities of the chromosome. Remember, we are talking about the karyotypes. That is, you can take out the amniotic fluid over there, culture them, and then you're going to check for the chromosomal analysis for the karyotyping, and also if there's any diseases the fetus is undergoing. And genetic engineering is one more, that is how your DNA and RNA is going to be introduced in the genes. And of course, the gene therapy, that is how you can use this in the gene therapy. The first cells removed from the patient, and the lacking or the functional one is induced, and then you're going to again reintroduce them, and these cells can reproduce that particular protein of interest which is being devoid in otherwise affected patients. So another method by using this viral vector is a functional gene is inserting the genome into the viral vector and allowing them to infect the patient. Thereby, that particular viral is going to express that particular protein of interest. Drug discovery, of course, animal cells are used to study the toxicity of the new drugs, and cell-based drug discovery. That is, for example, when a drug has to be validated from the day one to when it has to come onto the counter. So first thing is they are going to do the research for 3 to 6 years. Then preclinical studies, that is clinical study on the animals has to be done for 1 year. Then clinical trials for 4 to 7 years. Then after that, that will be reviewed and approval for another 1 to 2 years. So see the amount of years that goes in drug discovery and validation process. So yeah, so these are the different types of applications where you can use this cell culture for proteomics, for intracellular activities, cellular flux, that is RNA processing, pharmacology, cell-cell interaction, and of course the toxicology, tissue engineering, genomics, and immunology. So yeah, so this is what I wanted to talk about in propagation and maintenance of cell culture. So hope you have understood this class, and I hope you have understood all these topics over there. Any doubts, ma?
No, ma'am. Understood, ma'am.
Understood, ma'am.
If at all you have any doubts later in your while you're going through that, you can get back to me, and you can ask any doubts. You can post them. So just go through this topic and come back into the next class, and you can also discuss all these points. Okay.
Okay.
Yeah.
Okay, madam.
Thank you.
Good
Thank you,
afternoon,
madam.
ma'am. Thank you.
Good afternoon.
Thank you, ma'am. Thanks.